Good morning to all. Today we are discussing about screening methods. What is screening and why we want to know the meaning of screening. So, first we want to know screening methods. In previously, the screening is a basic, fundamental, and initiative process. In this process, mainly used in industrial purpose to isolate the production strain or fermentative strain. It's the initial process. It is only method is enough to isolate the organism means it's not enough because it's a elementary method, it's a basic method. So 100% purity of process is not to get in this process. So we want to pure further process. We want to pure 100% production strains, 100% giving yield giving strains isolated by secondary screening methods only. So um, uh, first the industrial microbiology, why we do these screening methods. So industrial microbiological field is defined as the study of larger scale and profit motivated productions of microorganisms of their products. It is a direct use of this industrial process. Industrial microbiology is primarily associated with the commercial exploitation of microorganisms. The commercial exploitation of microorganisms while doing this screen, we want to isolate a production strain or fundamental basic strain microorganism. The production strain 100% exploited in commercial product formation. That's why commercial exploitation of microorganism and involves process and product that are of major economic, environmental, and social importance throughout the world. So the first day we want to produce a production strain. In the strains, next to go enter the a fermentation process. So next day we want to know what is fermentation. So fermentation is used to describe anaerobic process to convert starch grains into alcohol. A process still used in first generation biofuel. Not only biofuel, during the fermentation, we want to produce multiple choice of product. That means antibiotics, amino acids, vitamins, vaccines, etc. etc. These are all the products produced in industrial field, instead of using microorganisms. That microorganisms are isolated by the screening methods, that is primary and is secondary screening. Next, next, next we enter the production strain. What is production strain? Production strain is nothing but a recombinant yeast or some other material, microbial agent that has been genetically engineered. So why we need to genetically engineered means the wild type of organism does not give too much of yield. That's why the organisms are genetically modified for the purpose of higher yield production, to make a desired compounds or products by means of the fermentation process, that is called as a production strain. The particular production strain having the ideal characteristics. What are all the ideal characteristics is found? First, we want to discuss able to grow on relatively cheaper raw material. The cheaper raw material, the investment of the investors must having the mindset is the raw material very cheap and locally available material. We want to expect that. That's why able to grow relatively cheaper raw materials. Well, first way is easy to cultivate, and or another way is easy to purchase in cheaper manner. Second point, grow well in an ambient temperature, preferably that is 30 to 40 degrees Celsius. And main ideal characteristics is high yield quantity of the end product, and also the concentration, minimum reaction time. So there is no extend time more than time for the reaction time don't exceed a limited time. Equipment used in fermentation process and also the stable biochemical characteristics. If it is we change pH or temperature, the organism does not change in biochemical character. Next point, yield only in desired substance, does not exceed undesirable substance only. It will give 100% of the desired substance. That's why we choose a production strain. Next one, possess optimum growth rate so that it can be easily cultivated larger scale. These are the ideal characteristics we keep in our mind while choosing the production strain. Next, we enter the screening method. What is screening? Screening is a procedure or basic method for to isolate reduction and separation of microorganisms from the mixed population. The mixed population science is it is otherwise called a soil, that is a heterogenic population because having multiple choice of microorganism. The soil having a microbial diversity. That's why we isolate the organism from mixed population. In this process is called as screening. Next is a protocol. So first we want microorganism or production strain or fermentation strain. So we go into the sources. What are the sources are found in our in and around the environment? First, food means milk and cheese product, etc. These are all the some of the food material may give the production strain or fermentation strain. So second source is environment, is the best source, cheapest source. So air, water and soil. Air, it will provide a certain amount of certain type of organisms, and also water it will give a certain group of microbes, but the soil only give microbial diversity, having the microbial diversity and give lot of microbial populations. That's why we are prefer the soil content. Next one is compost and other sources. This three kinds of sources will give the production strain or fermentation strain. So the three sources we want to use the method screening. The screening method is further divided into primary and secondary screening. Primary is a basic one, secondary is a final stage. So primary screening is a basic. So types of screening is having two types. One is primary screening, second one is secondary screening. Type of screening, they are primary screen having four types. So first dyeing, use of indicator dye, it will produce organic acid producing microorganism. And a crowded plate method, it will produce amino acid producing microorganism. Third one is oxonotrophic technique. It is extracellular metabolites producing microorganisms. At the defined media, enrichment culture technique, we want to use the defined media. It will enrich the culture and it will produce a lot of products, and that means to enzymes producing organisms, enzymes products. Secondary screening is here it's also having three types of methods. One is gain colony methods, filtration method and a liquid medium methods. So primary screening having four types. One is organic acids by dyeing, organic acids and amine by dyeing method, amino antibiotic by crowded plate method, extracellular metabolites by oxonography method, enrichment production, enzyme production by enrichment culture method. Next, we enter the important things. Before to enter the primary screening, some important things to be considered while screening. First one is sources, second substrate and deduction. Source already we talk about soil, air, milk, compost. These are all the sources to give a sample by the way of screening methods. Second one is substrate. Without substrate, the any organism does didn't live. So the nutrient and growth factor has to be supplied for the growth of desired microorganisms. So first priority is given due to sources. Second one is substrate. Finally, the microorganism deduction, choice of deductions, a proper isolation reduction of desired microorganisms is very important. So the primary screening, so it may be amino acid acids and amines. It's for amino acid amines producing organisms by indicator dyeing method, but the amino acids and amines does not produced by crowded plate method. The crowded plate method only give you antibiotic producing organisms, and indicator dye is produce organic acids and amines, and the extracellular metabolites produced only by the oxonography. So no other method does not produce any other organism. That is a choice of reductions of isolations of microorganisms. Next, we enter primary screening. Primary screening is defined as detection and isolation of desired microorganism based on its qualitative ability to produce the desired product of amino acid, antibiotic, or an enzymes. Here the process, desired microorganisms generally isolated from the natural environment, that is soil, contain different species, several different species. Sometimes the desired microorganism has to be isolated from large population of different species of microorganisms. This these are all the important points we need in primary screening time. Next, next we go for the next slide. Primary screening, first method, crowded plate. In this method employed for detecting microorganism while capable of antibiotic producing organism. Here this technique started with the selections of natural substance like soil or other source consisting of microorganisms. First, we start in serial dilution of the sources made. Suitable the serial dilution is chosen which is able to produce 300 to 400 individual colonies were plated and an agar plate after incubation. Such a plate is called as a crowded plate because a crowded plate in sense crowded of individual colonies were isolated. The antibiotic producing activity of colony indicates no growth of any other bacterial colonies, that is clear colorless area around the antibiotic producing microorganisms. This region is called a growth inhibitory zone or the clear zone of incubation. These are all the points we are kept in our mind while doing the crowded plate method. Next slides indicates axonography method. Here, this technique is employed for the detection and isolation of microorganisms capable of producing certain extracellular substance such as growth stimulating factor like amino acids, vitamins. Definite growth requirements for the particular metabolite used in this method. For this purpose, spread on the surface of the sterile agar plate and allow the growth isolate colonies after incubation. Finally, to get such colonies or isolate such colonies in means extracellular substance producing organism is isolated, purified, and stock cultures are prepared, which are used for the further screening process. Next, we enter the enrichment culture method. In this method, generally employed to isolate those microorganisms that are very less in number in soil sample and possess specific nutrient requirement, because enrichment meaning is to enrich the substrate, so excess nutrient it will added to the media. They can be isolated if the nutrient supply required by them is incorporated into the medium or by adjusting the incubation condition. The final primary screening method, indicator dye technique. Here the microorganism capable of producing acid and amines from natural source can be detected. Using this method by incorporating certain pH indicator, that is neutral dye, neutral red or bromothymol blue into the nutrient agar medium. In this, changes in the color of the particular dye is the vicinity of the colony. It will indicate the ability of colony to produce organic acids and bases. This is the indicator dye. This is the final methods in primary screening. Next, we enter the secondary screening. Secondary screening, the primary screening does not give a detailed information about the organism. That's why we need secondary screening. So primary screening help in the reduction and isolation of microorganisms from the natural substrate that can be used for industrial fermentation for the productions of compounds of human utility, but it cannot give the details of the production potential or yield of the organism. Such details can be ascertained by the further experiment. That's why this is known as secondary screening, which is provide broad range of information pertaining the organism. The organism uh, while doing the secondary screening, the organism give the information about ability or potentiality of the organism to produce metabolites that can be used in an industrial organism, quality of the yield product, fermentation process that is able to perform, elimination of the organism which is not industrially involved. These are all the points while doing the secondary screening. They are get it from this method. Finally, to evaluate the true potential of the isolated microorganism, both qualitative and quantitative analysis is generally conducted. Next, evaluations of potentiality of the microorganisms. Microorganisms isolated in the primary screening are critically evaluated in the secondary screening so that industrially important and the viable potential can be assured. They include the sum of the points. One is to determine the product produced by an organism's new compounds or not. Next one is it should mm, detect whether the isolated organism genetically stable or not. So next, reveals suitability of the medium or its constraint chemical is to the organism yield potential, the chemical stability, physical property of the product. These are all the points they are given by the secondary screening method while doing the secondary screening method. Next, it should select industrially important microorganisms and discard others which are not suitable for the fermentation industry. Determine the economic status of the fermentation process undertaken by employing newly isolated organisms. Next, methods of the secondary screening. The secondary screening gives very useful information pertaining to the newly isolated microorganism that can be employed in fermentation process of commercial value. These screening tests are conducted by using petri dish containing solid media or by flask or a small fermenter containing liquid media. Each method has some advantages and disadvantages. Liquid media method is more sensitive than agar-plate method because it provides more useful information about the nutritional, physical, and production response of an organism to actual fermentation production condition. This is the useful information while pertaining the secondary screening. Next, types of secondary screening methods. One is gain colony technique. Another one is filtration technique. Third one is liquid medium. These three methods comes under the category. Second is screening method. Three methods to use to isolate 100% yield giving microorganism that micron genetically stable, physically stable, biochemically stable. It's give only 100% yield, none of other products. These are all the techniques very useful in secondary screening methods. Next is thank you.